Hello everyone. Uh, in this bioinformatics practical, we will be retrieving protein motif information from Prosite database. So in the platform, a practical is the procedure for the practical is given, and the website address is also given here, prosite.xpressy.org. If you click on this website, you will open this homepage of a Prosite. You can go through the website, database of a protein domains, families, and functional site. That is Prosite. So here you have a search box uh, where you can enter the protein ID, or you can go to the scan mode in which the FASTA sequence or the protein identifier like a UniProt accession number, etc., can be pasted directly here. And the details about the or motif related information about the protein can be retrieved from this site. So from where you will get such a sequence like a FASTA format or a UniProt KBID, it is normally from the UniProt website. You can go to the UniProt KB knowledge base website, and if you are interested in insulin, you can search for insulin. Select the database as UniProt KB and search. So you will get the results. So this is the insulin growth factor. It is not our interest. Insulin like. This is insulin. Homo sapiens insulin with 1 ton. So if you are trying to download a protein or insulin, this ID will be sufficient. This is the UniProt accession number. Or you can get the details about the protein, and at the bottom of the page, after all the annotations, you will see the sequence, and the FASTA format of the same protein can be downloaded using this provision that is provided here. Here, this is the FASTA link of that one isoform, and this 1-ton amino acids can be downloaded from this location. So in this website, you require only the protein ID that is provided, say here. This ID can be copied, and you can paste it here, then scan. Alternatively, you can get the download the FASTA file, and that sequence can be provided in the box. So once you get the results, you can see that this is the human insulin. From that identifier number, the 1-ton amino acids along the insulin protein that is located, and full details about description about the protein and the source is given here. This is the actual sequence. And now when we come down, you can see that this is the hit patterns. Okay. So here you don't have any specific motifs that is present here. It's a very small protein, and the active sites or other sites were not present in this protein. Whereas if you go for another protein, for example, say a larger protein, this one, you can again give the identifier number here or paste the sequence, and scan. The sequence is given, and here you can see the different motifs of that protein provided here, and the additional details about that motifs and the sequences, etc., and from where it starts and ends, the amino acid, starting amino acid and ending amino acid, and their score, etc., were also shown at the bottom. And now when we come to the top, by clicking on this one, for example, the sequence is shown here. If you click on the, sorry, if you select, say, okay, here, by changing the cursor over this domain graphical interface, the highlighted region will change. So these are the sequence that constitute this FN3, and this FN3 is from this one, and this protein kinase domain is from this region. So just by moving your cursor over this motif, you can have that sequences highlighted in this page, and also you will have other additional information about each and every motif that is available here. So motif related information of any protein can be retrieved from this website, that is Prosite.